like that?
Yeah.
Yeah, I had them crossing over though.
Yeah.
So you have them crossed over or synapsed? So then at metaphase 1, what should I have in that?
2 X's.
Side by side. Homologous pair.
I should have homologous pairs
Nice.
aligned at the metaphase plate. Do we agree?
Yes.
Then we move on to anaphase, and what do we have?
They're not taking the opposite side.
So I have one there and one there.
Yeah.
Okay. And then we need a new slide, right?
Little A's.
Little a's. Or are they all lowercase a?
No,
No,
they're big.
Some are uppercase a?
Yeah.
Okay.
I remember this.
On your chart, if I had, before prophase 1, if I had drawn interphase, what would have been in a cell? Say this is the interphase. So before DNA synthesis, before replication occurs, what's in each of these cells?
2, or no.
2n equals 2. It's still diploid, but are they X's or are they lines? So what color would my lines be? One green and one weird yellow orange? Okay. Here's what I want you to think about. On your green chromosome, what letter is it? Lowercase a or capital A?
Lower.
Oh, so everybody's is different. How many of you are like, but on my green one I have an uppercase A? And on my green one I have a lowercase a? On your green chromosome in prophase 1, are they both uppercase or both lowercase, or are they mixed?
I have.
How many of you have the same? They're both either, it's both uppercase or it's both lowercase. Anybody in here have one that is different? Just on the yellow.
Oh.
So just
okay.
on yellow or just on green?
Just that ball.
Just on yellow or just on green?
Oh. Okay.
So why does it make sense?
Because if it, let's say, because if it's like before it duplicates, before it replicates, it has to make an exact same copy of itself, so it's automatically going to be both of the.
So they both should automatically be the same because it's just each homologous or each sister chromatid is just a copy of the thing it's attached to. So on your cheat sheet, your summary sheet, go ahead and draw that interphase cell in. How about this? I'll give you a moment to draw in all of your answers on that cheat sheet, summary sheet. And some of you will have to write lowercase. My example here. Looking at my interphase or prophase 1 cell, I labeled them all. Are they all in the same spot on my homologous chromosomes? Like same location? What's a gene again? All right, flip over your summary sheet and go ahead and write that definition for me in the spot that says define gene.
What is it?
What is a gene? Section of DNA that presents or represents a trait that, so it's a section of DNA that codes for protein. So all we have is a section of DNA here that codes for protein.
What if it doesn't get the protein?
Every gene, so every organism that is born as a healthy, normal organism is born with every recipe for every protein. Now, I have two different letters here that you can hardly tell because I wrote so very tiny. So there's my lowercase a, there's my uppercase a.
So, if they cross over, like, does that mean that they just make like a totally different trait that mom and dad don't have?
It's not that they're going to make a different trait. What's going to happen is if they cross over, so remember crossing over in synapsis? If my two right here were to cross over and synapse, I would have one chromosome with a lowercase a at the bottom and one chromosome with an uppercase a at the bottom. And reversed here, there'd be an uppercase here and a lowercase there. So it's just a swapping of recipes. I started with two different copies, right? An uppercase A and a lowercase a. But they're both the letter A. Agreed? So let's see, what trait can this one represent? When you eat beets, if you eat beets and then pee the color pink, you are beta-uric. It's an actual genetic trait you can inherit. So if you eat beets and you pee pink, it's because you inherited a gene that lets you pee pink when you eat beets. And some people inherit a gene, that same gene, but a different copy, and they don't pee pink when they eat beets. Their pee remains yellow. Beets, B-E-E-T-S. So let's just say that's what this is. This individual got the I can pee pink gene from one parent, and the I can't pee pink gene from another parent.
That'd be fabulous pee.
Then they, throughout the course of their life, went through puberty and were going to make gametes. So interphase happens, DNA synthesis happens, and they go to form gametes. These are just exact copies of this one gene that I had, right? Of this one chromosome. Exact copies. And this is an exact copy of that chromosome. Those are the exact copy. When we So we left off on the last slide with what, did I have green on the left or green on the right? So what should be in my left cell here?
Green X.
Aligned where?
Center.
Okay. And in the right cell?
Yellow.
Okay. Now what should be in my 4 cells after that?
Green line. Just like the yellow one.
And then, depending on whether you and your partner agreed to be male or the female, if you agreed to be the male, you should transfer those into the male gamete, right? And if you agreed to be female, you should transfer those into the female gamete. But hey, how is this any different than what we've already done?
It's reviewed.
Wait.
What's different about this?
Oh, it has the beads on it.
Has the beads on it. What's written on those beads? Because you had lowercase a, but mine, for the sake of demonstration, I said they were all capital A's. And then once you've done that for the one chromosome, go back and do that for the other one as well. So if you've done the green already, go back and do that for yellow. those letters represent?
Gene.
Stuff.
Great. Gene.
What's a gene? Then. So when we come to make the gametes down here, one gamete got a I can pee pink. Another gamete got I can pee pink. Another gamete got I can't pee pink. And the other gamete got I can't pee pink. Do you see how those got divided out because of meiosis? So the four possible things that could have been passed on, the four potential gametes that got made, there's actually only two that are different, right? They either could give a gene to their child that says, yes, you can pee pink, or they give a gene to their child that says, no, you can't pee pink. Still with me? Same protein product, right? So it's still the same gene. It controls pee color. It's a real gene. I'm not making it up. Don't believe me? Go home and eat some beets.
I'm pretty sure it happened as well.
You should probably try it. It's very entertaining.
Well,
How likely is it that you'll pee pink? It is the dominant trait.
Isn't that something?
The fact that none of you know it means that too few of you eat beets. That is horribly upsetting. How much beet do you have to eat to pee pink? Not a whole lot. Beets are pretty powerful.
Yeah, like when we get
So like both parents could pee pink and would like, is there a way that the kid can't pee pink?
That's an interesting question. That's why we're doing this.
Wait, what do you mean more dominant? Do you mean the person that like?
Oh, we'll get there. We'll get there.
dominant.
It's coming. Not necessarily.
Well,
Not necessarily. That's what, yeah, see, we're getting there.
dominant.
The four gametes I made, capital A, lowercase a, lowercase a. Anybody in here make all capitals? Did anybody in here make all lowercase? Is everybody else half lowercase, half uppercase? Okay. So your question was, is it possible for parents who are all lowercase to make a child, or who can't pee pink, is it possible for them to make one who pees pink? Was that the order that you asked the question?
If they could both pee pink.
So those beads and those letters on the beads, those beads represent a gene. And are they all located on the same place on those chromosomes? Do they all occur at the same section of chromosome? So let's go back and look. The letters on the beads represent, or the bead itself represents a gene. The letter, whether it's capital or lowercase, represents the version of the gene you get. Some genes have multiple versions. Some genes only have two versions. In the instance of the P beet gene, there are two versions. They're either pink or not pink, and you have no other options. So from your parents, from one parent you got a pink or not pink, and from the other parent you got a pink or not pink, and you have no other choices there. Versions of genes have a very special name. We call them an allele, A-L-L-E-L-E, an allele. So here we have two alleles, either capital A or lowercase a. There are two choices for that one single gene. Now if 2n equals 2, what does n equal? Okay. So if 2n is 2, n equals 1. What genes were transferred to your gametes? So your 4 gametes, you should have made 4 down at the bottom. What are the possibilities? And you can write 4, just write, like, say, I would write, based on mine, capital A, comma, lowercase a, comma, lowercase a, because those are the 4 gametes I made. Some of you yesterday drew a picture on your "this is what I know about genetics" that looked like this?
Punnett square.
It's a Punnett square. And what most students misunderstand about Punnett squares, I think there's two ends. Oops. Most people, most students, most people don't understand where the letters that we write on the side come from. The letters that you write at the side of the Punnett square, for those of you, how many of you have done Punnett squares before? You've seen a square in genetics before? Okay. The square, the letters on the side come from the alleles, the versions of genes you would have gotten from your parents. Now going back to mine, I said that I would have done uppercase A, comma, uppercase A, comma, lowercase a, comma, lowercase a. Those are the four I made, we all agree? So on my side of the Punnett square, at this box, I'm going to write uppercase A because that's one of the genes that or one of the alleles, one of the gametes I would have made. I also could have made a lowercase a. I don't need to make an extra 2 boxes here because I really just passed on the same gene twice, right? You agree uppercase A is still uppercase A? So why do twice the work? Write it once. But here's the deal. This is only mine. This is only my DNA. Is this whole thing going to happen that I pass it to offspring with only my DNA? My offspring is also going to get whose DNA? Yeah, my husband's DNA, right? So let's just say my husband is the same as me. He made the same 4 potential gametes. So he made an uppercase A and a lowercase a. If one of my gametes that was an uppercase A is fertilized by one of his gametes that was an uppercase A, what 2 genes did our children, or 2 alleles did our children inherit, or child inherit? Both uppercase A's. So they will both possess the trait in question. They will both, so they pea pink. When you have the same allele, we have a name for that. We call it homozygous. That means you got 2 versions that are the same. So in this case, we are homozygous, capital A, capital A. Let's take a look at the child that would occur from the fertilization of the next 2 gametes. So we can either go down or right. What one do you want to do?
Down.
Okay? So that's the one that the child would get from my husband, right? What one would the child get from me from that gamete? Lowercase a. Now you'll notice I wrote the uppercase first. Whenever we do a Punnett square, we always write the uppercase first. Collect like terms, write uppercase first. So that child would have gotten a yes, I can pea pink from my husband and no, I can't from me. So wait, those alleles aren't the same. Can the kid pea pink?
It depends which one is dominant. Is that what you just said?
When you get 2 different alleles for the same trait, so the trait in question, pea color, when you get 2 different alleles, capital A, lowercase a, we call that heterozygous. Hetero means different. So they are heterozygous. Now the question is, can this child pea pink? And we said it depends on which one is dominant. How about this person up here then? Capital A, where capital A means yes, I pea pink. Will that person pea pink? Yes. Here, capital A, lowercase a. Capital A means yes, I pea pink. Lowercase a means no, I don't. And capital A is stronger It will always be expressed. So can that individual pee pink?
Yes.
How about this individual?
Yes.
And this one?
No.
So there's only one individual who will not pee pink from this union of parents. Right? So to answer your question, can two people who pee pink make a child who doesn't pee pink? If the two people are heterozygous, so they have to each have a recessive allele to make sure that I have a child with two recessive gametes, or two recessive alleles. Make sense? On the bottom of your sheet, I do believe the next set of instructions says something like, if the gametes in your lab partner's diagram combined with the gametes from your diagram, what would all of the potential combinations be? How do you figure that out?
That's where.
A Punnett square. So in that space, make a Punnett square. So let's define that word. Dominant and recessive. Most people assume it means with the frequency with which that trait appears. Oh, it happens more often. That trait is dominant. That's not actually what it is. Remember, there are two different recipes for a protein. And all it means is that individual is going to use both recipes to make that protein in their lifetime. They get two recipes, they use both. Sometimes one recipe is stronger than the other. So here's my example that you'll totally understand if you've done this before. How many of you have added mint extract to a recipe ever? Mint, the mint flavoring. So there are chocolate chip cookies. Agreed that a chocolate chip cookie is a thing, right?
Yeah.
And there are multiple recipes for the same thing. So I can use one chocolate chip recipe that is just a chocolate chip cookie, nothing special added, just like old school chocolate chip cookie. And I can use a chocolate chip cookie recipe that makes a mint chocolate chip cookie by just adding a couple of drops of peppermint flavoring. Right? It's possible. Still making a chocolate chip cookie. If I store both of those cookies in the same container, what's going to happen to my non-chocolate or my non-mint cookies? Yeah, because mint is a lot stronger than the absence of mint. So all of the cookies in the container are going to taste like mint. So when you use different versions or recipes of genes, sometimes one masks the other one being there. You can't tell it's there, you can't see it, you don't know it's there because the strong one overtakes the weaker one and you don't know it's there. So in this instance, the one that masks the presence of another gene that is always expressed, you always see it, that's the dominant one because the protein that gets made hides the presence of the other protein. You don't know it's there. So my poor non-mint chocolate chip cookies, even if you ate those cookies, you wouldn't know because the mint extract is so powerful, it will make the non-mint ones taste like mint. That's why I don't use it often because it's slightly overpowering. Does that make sense to you guys? So dominant genes are the protein that's always expressed, the one that we always see, no matter what. And recessive is the protein who can be masked by the other. So it's masked by a dominant one. The only time we would ever see the recessive trait then is if you got two recessive alleles. So the only time I would ever make non-mint chocolate chip cookies is if I followed two recipes that did not involve mint at all. So going back to